Hello everyone, I am Reshma Kini and we'll be discussing the topic of apoptosis in this video. So at the end of this video, you should be able to explain the role of apoptosis in human health and disease, explain its etiology, that is the cause of apoptosis, mechanism, that is how apoptosis takes place, and the morphology, that is how an apoptotic cell will appear. So when a cell undergoes injury, the injury is either reversible, that is the cell after getting injured will revert back to its original state and function, or when it is irreversibly injured, it will undergo cell death. So when a cell is irreversibly injured, it can choose or it can undergo cell death in many different ways. So the most common one that you have already read about is accidental cell death, that is the necrosis. And then we'll be seeing today about what is apoptosis, that is the programmed cell death. Apart from these two, there are several other mechanisms like necroptosis and ferroptosis that the cell can choose to die by the way of. So unlike others, apoptosis is controlled, energy dependent and highly regulated. What it means is that it follows a specific sequence of steps. Hence, it's also called as programmed cell death. The word apoptosis comes from Greek words apo, that is away from, and tosis, which means falling. So put together, apoptosis translates into falling off, maybe like a leaf of a tree. But how I would like to compare these two is in Hollywood fashion. That is, a necrosis is like a bomb blast. When there is a bomb blast, you have a lot of damage, a lot of chaos, and a lot of you can say people or organisms, whatever are there, or cells, these represent cells, dying off. Okay, so it is uncontrolled and it is messy, and usually it is followed by chaos. Chaos in this case represents the inflammation. Okay, so for necrosis is uncontrolled, it is chaotic, and it is followed by inflammation, where we have the white blood cells rushing to this area to check what has gone wrong. Okay, so an area of necrosis is usually surrounded by inflammatory cells because it is chaotic. Whereas apoptosis is something like a sniper attack. You have a lot of people and a lot of things, but a sniper takes out only that individual, or in this case only that cell which needs to undergo cell death. Okay, so there's no chaos here. Okay, so whatever is the cell that is dead, that is quietly come and it is removed off by the circulating macrophages. Okay, so in comparison to necrosis, which is messy, we have apoptosis, which is very quiet, which is very targeted and very specific. So when and where do apoptosis happen? So apoptosis happens normally in the body when the body needs to eliminate older cells or damaged cells. It helps in maintenance of tissue homeostasis during development. That is, while making individual fingers of hands by removing the webs from between the toes. That is, in case this did not happen, we would not have had individual toes like I am showing in the picture. We would have had feet like that of a duck. It eliminates cells which are harmful. Now, what are the cells that are harmful to the body? Self-reacting lymphocytes. So lymphocytes that react to our own antigen have to be gotten rid of. Otherwise, if they do survive, they can cause autoimmune disease. Tumor cells which are generated in the body also need to die, and they are also made to die by apoptosis, and also cells that are infected by viruses. So dysregulation of apoptosis, that is, if apoptosis did not happen, it can lead to cancer. It can, that is, the cells having mutations within them, they learn to avoid apoptosis to give rise to cancer. Or it can give rise to autoimmune diseases, that is failure of apoptosis in immune cells which react to one's own antigen. It can also give rise to neurodegenerative disease, that is the cells of central nervous system, they undergo untimely apoptosis, and these are called as neurodegenerative disorders like Parkinson's disease and Huntington's disease. So in summary, apoptosis can be physiological or pathological. It can be beneficial or harmful depending upon where and when it is occurring. And you also need to know apoptosis because there are drugs now which can target this process. So you have drugs which can induce apoptosis, for example in tumor cells, or drugs which can hamper apoptosis. You would need this in case of treatment for neurodegenerative disorders. So what triggers apoptosis? Okay, we know now that apoptosis can be both physiological as well as pathological. So what triggers it? Who tells the cell that yes, you have to now undergo a programmed cell death? So it can be because of presence of signal which is received by the cell. Okay, so there is presence of something, presence of a signal for apoptosis received by the cell, which can come from within the cell or it can come from outside the cell. Or it can also be because of absence of something like growth factors which sustain a cell. So apoptosis can happen in two cases. One is because of a signal or because of absence of growth factors. Now when we come to apoptosis because of signals, it can come from within the cell, in which case we call it the intrinsic pathway because the signal is coming from within the cell, or we can call it apoptosis can happen using extrinsic pathway because here the signal is coming from outside the cell. So initiation phase, there are two phases in apoptosis, the initiation phase and the execution phase. So initiation phase can happen either by the intrinsic pathway or the extrinsic pathway, whereas for execution phase it is a common pathway. It is very similar to the one that we see in coagulation, where we have an intrinsic pathway, extrinsic pathway and then a common pathway. So to execute apoptosis, we have certain enzymes called endoproteases. Okay, and these are called as caspases. There are two groups of caspases, the initiator caspases numbered 8, 9 and 10, which take care of the initiation phase, and the effector caspases number 3, 6 and 7 that take care of the execution phase. So let us see about the phases of apoptosis. I told you that there are two phases, that is initiation phase and the execution phase, and I also told you that the initiation phase, in the initiation phase the signal for apoptosis can come from within the cell or from outside the cell. So from within the cell, when it happens, we say that the initiation of the apoptosis has occurred through the intrinsic pathway. The intrinsic pathway of apoptosis is regulated by two groups of proteins. The ones that prevent the apoptosis from happening, that is the anti-apoptotic proteins, which is the BCL2, BCLX and MCL1, and the pro-apoptotic proteins which encourage the cell to undergo apoptosis, that is BAX and BAC. And these two groups are maintained in balance by what is called as sensor proteins. So when a cell gets irreversibly injured, it could be because of radiation, it could be because of chemotherapy, or maybe because of viruses or toxins, the sensor proteins sense that the cell cannot recover anymore and increase the amount of the pro-apoptotic proteins, that is the BAX and the BAC. So here you can see the intrinsic pathway. In this, the stimulus for death comes from within the cell when the cell is irreparably damaged. For example, the DNA is damaged irreparably due to chemotherapy or maybe radiotherapy or some other reason. So the sensor proteins sense this and they decrease the amount of anti-apoptotic proteins and increase the amount of pro-apoptotic proteins. So once BAX and BAC are increased, okay, what they do is they go towards the mitochondria and they create holes in the outer membrane of the mitochondria. So here they have created holes. So what this causes is it causes release of a molecule, these pink dots, which are nothing but cytochrome C. Okay, so once the cytochrome C leaks out from the mitochondria into the cytoplasm, it combines with what is called as apoptotic activating factor 1, that is these blue crescents, and then both together they will activate what is called as caspase 9. Now caspase 9 we already know is one of the initiator caspases. So once caspase 9 gets activated, it will activate the effector caspases, that is number 3, 6 and 7, and brings about the execution phase of the apoptosis. Okay, so this is the intrinsic pathway of apoptosis. Okay, started off by the sensor proteins which increase the amount of the pro-apoptotic proteins. So what happens in extrinsic pathway? So like I said, in extrinsic pathway, the signal for apoptosis comes from outside the cell. So signals come from where? They come from other cells which have what is called as a death ligand. Okay, so which are these cells? These are cells like T lymphocytes. So they carry on their surface what is called as a death ligand. So one example for a death ligand is a fast ligand. So this T lymphocyte, it engages with the target cell using its MHC receptor, okay, MHC receptor, and it will expose the death ligand to that particular cell. Now who is the target cell here? Usually a target cell is the one which is infected by any virus, okay. So in our body, whenever a cell gets infected by virus, it is recognized by the T lymphocyte using the MHC molecule, and then it engages with that particular cell using a fast ligand. So this particular cell, usually all the cells in the body, they express what is called as a fast receptor. So once this fast receptor is engaged, this particular cell, it will undergo apoptosis. Okay, we will see how. So this is the extrinsic pathway of apoptosis. So here we have the cell membrane. Okay, we have the fast receptors. We have fast receptor number 1, 2 and 3, and we have this middle fast receptor which has been engaged by the fast ligand, which is in maroon color. Okay, so the T lymphocyte has come and it has put its fast ligand into the fast receptor of this particular cell. Now once the fast ligand binds to the fast receptor, it will pull the adjacent fast receptors towards it and undergoes what is called as trimerization. Okay, it forms a triplicate, or it forms a triple bundle of 3. That is, it trimerizes, and in this process its intracytoplasmic domain. Okay, so this is the cell membrane, and this green color area is its intracytoplasmic component, which is called the FAD. Even the FAD will trimerize. Okay, so once the FAD trimerizes, what it does is it converts procaspase 8 into caspase 8. We know that caspase 8 is another initiator caspase which can activate the effector caspases, that is number 3, 6 and 7. So this is what happens in extrinsic pathway. The T lymphocytes or any other cell which carries the death ligand or the fast ligand, it binds with the fast receptor. The fast receptors will trimerize and activate the procaspase 8 into caspase 8, which will activate the execution pathway of apoptosis. So that is the second pathway. What is the third pathway of initiation of apoptosis is the perforin granzyme pathway. So here what happens is the cytotoxic T lymphocytes, they have this enzyme within themselves called perforin. Okay, then as the name suggests, perforins are the ones which can create holes into the opposite cell. Okay, so here the cytotoxic T cells recognize target cells using MHC molecules. They release perforins and form pores in the target cell membrane. So once they form holes in the target membrane, they release what is called as granzymes. Okay, these are enzymes into the particular cell. So this granzyme will later go into this particular cell and activate caspase 10 and finally caspase 3. Okay, so this is the perforin granzyme pathway. We have the cytotoxic T lymphocyte. It has created a hole in the cell membrane. Okay, this is by using perforin, and it has released these brown molecules, that is granzyme, into the particular cell. This granzyme has activated caspase 10 and finally it has activated the effector caspase number 3. Okay, so these are the 3 different mechanisms how initiation of apoptosis can happen. The intrinsic pathway, where the signal comes from within the cell, the extrinsic pathway where the signal has come from outside the cell, and the perforin granzyme pathway, which is also one of the signals where one of the signals is coming from outside, but we just don't call it an extrinsic pathway, we just call it a perforin granzyme pathway. So let us see what happens in the execution phase. So in the execution phase, it involves activation of effector caspases number 3, 6 and 7. Okay, these 3 caspases together with endonucleases, they break down the cell into small fragments called apoptotic bodies. We'll see how that happens. So here what happens is the initiation pathway is over and there is activation of effector caspases. So effector caspases by themselves will go and start breaking down the cytoskeleton of the cell. Now what is a cytoskeleton? It is something that holds the cell membrane to the nucleus. Okay, it binds the cell membrane to the nucleus as well as maintains the shape of the cell. So when we have the effector caspases breaking down the cytoskeleton, the connection between the cell membrane and the nucleus is lost. So the cell membrane is now free to move away from the nucleus. Okay. The other thing that the effector caspases do is they activate endonucleases. Okay. Nuclease is an enzyme which will break down the nucleus. So this endonuclease will break down the DNA, okay, DNA into 180 to 200 length base pairs. That is, it breaks down the DNA in a regular fashion, each of the DNA fragment being of 180 to 200 base pair. This is the most characteristic feature of apoptosis, okay, breaking down of the DNA into 180 to 200 base pair length. So what happens here is, once the cytoskeleton is broken down, once the nucleus is broken down and the cell membrane is allowed to move freely, okay, it will move away from the nucleus and begins to form small bodies all by itself like this. So this cell, because the cytoskeleton is no longer holding it together, it has broken down into or formed these small things called apoptotic bodies. Now these apoptotic bodies are just smart enough to express on their surface, in green that you can see, certain molecules which make it very tasty for the macrophages who are just roaming around there. Okay. So once the cell undergoes apoptosis and forms the apoptotic bodies, it expresses on its surface these green molecules, okay, which makes it very tasty for the macrophage who come and they just eat up the apoptotic bodies that are lying there. Okay. So once the same thing is depicted over here, the cytoskeleton is broken down. So the cell membrane moves away from the nucleus also begins to break down, and then there is the formation of apoptotic bodies, and these apoptotic bodies, they express tasty molecules on their surface and they become a fodder for the macrophages that are roaming around there. Now the important thing to note here is, there is no bursting of the cell and releasing of its content into the environment, which happens in necrosis. Okay. So in necrosis, there is damage to the cell membrane, which is making the cell burst and release its content to the outside, and that causes inflammation. Here, because the cell contents are still bound by the membrane, okay, no chaos is happening, no release of content is happening, and there is no chance for the other inflammatory cells to come there and do some sort of chaos there. Okay, so this happens very quietly. The cell quietly breaks itself down into small apoptotic bodies. The macrophage quietly comes there and it quietly eats up these apoptotic bodies without any fuss. So that completes the mechanism of apoptosis. We have seen the initiation phase, which can happen by intrinsic pathway, extrinsic pathway and perforin granzyme pathway, and the execution phase, which is done by the effector caspases, finally leading to development of the formation of the apoptotic bodies, which are quietly removed by the macrophages. So let us see what morphology of apoptosis is like. That is, what does apoptosis look like in the microscope. So in the light microscopy, if this is a cell, in the light microscopy, the cell appears shrunken. So when a cell is undergoing apoptosis, it shrinks. Okay, it shrinks, and because it shrinks, the cytoplasm begins to look more pink than the normal cell. Along with that, the cell organelles, they also begin to come towards each other. The nucleus also changes its appearance from having a chromatin which has some kind of texture that you can see here. It shrinks into a small condensed ball and loses all its internal structure. So this is called as karyopyknosis, okay, shrinking of the nucleus. And then this, because of the action of the endonucleases, if you remember, where the endonucleases breaks it into base pairs of 180 to 200 length DNA, the nucleus fragments, okay, and this is called karyorexis. And this is where the cell begins to develop outpouches like this and form the apoptotic bodies. So this is what it looks like in light microscopy. There is shrinkage of cells, there is karyopyknosis, there is development of the cytoplasmic webs, formation of apoptotic bodies and karyorexis. So in the electron microscopy, there is characteristic feature. Okay. Along with the visibility of the cell shrinkage and the organelles that begin to get tightly packed, there is condensation of the chromatin under the nuclear membrane. So this is the nucleus and there is condensation of the chromatin under the nuclear membrane in this half moon shaped structures. Okay. And in the electron microscopy, the formation of the blebs is also very evident, and here is where the blebs are getting pinched off and formation of apoptotic bodies is happening. So all this is visible under electron microscopy, but the most important thing to note is condensation of the chromatin. Okay, this is chromatin. It condenses under the nuclear membrane in these half moon shaped structures. This cannot be visualized in the light microscopy. So in summary, apoptosis is one of the ways of cell death. We know that there are other mechanisms, and this is one of the mechanisms. Because it is highly organized, we saw a series of steps, okay, both in intrinsic, extrinsic, porphyrin granzyme pathway as well as execution pathway. There was a specific sequence of steps which was maintained, and because of which it's called as programmed cell death. We also know that caspases are the ones which initiate as well as execute the apoptosis, okay. And then in the initiation phase of apoptosis, we can see that it can occur by three pathways, that is the intrinsic pathway, extrinsic pathway and porphyrin granzyme pathway. We know that the initiator caspases are number 8, 9 and 10, and the effector caspases are 3, 6 and 7, and these together break down the cytoskeleton and DNA of the cell. So once the skeleton is broken, the cell membrane undergoes blebbing. It forms apoptotic bodies which make themselves tasty to the macrophages, and the apoptotic cells get eaten by the macrophages, and because the cells don't release their content in the surrounding environment, there is no inflammation. Following this, we saw the light microscopic changes, that is cell shrinkage, followed by karyopyknosis and karyorhexis of the nucleus, and how the nuclear material gets condensed around the nuclear membrane in electron microscopy. So that completes apoptosis for you, the etiology, the mechanism of apoptosis and the morphology, that is appearance of apoptosis. Thank you for your patient listening.